Okay, I think we're ready to start. Welcome to the EMGS webinar on AOPs. What are they and how the EMGS can be involved? This call will be recorded, and we would ask everyone to please mute your mics at the very beginning of the call. And if you have questions during the call, please use the chat box, and we'll try to address those at the end of the presentation. Thank you.
Thanks very much, Bob. So welcome, everybody. This is Carol Yauch speaking. I'm Vice President of the EMGS. And my day job is a research scientist at Health Canada. My lab is interested in the application of new methodologies in genomics in risk assessment. And I'm sitting here today with Dr. Jason O'Brien from Environment and Climate Change Canada. Jason and I have been working together for many years. Jason's job at Environment Canada is actually very similar to my job at Health Canada. Our research foci are highly aligned. And one of the things that we do together is we serve as Canadian delegates to the Organization for Economic Cooperation and Development, so the OECD. And specifically, we serve on their Extended Advisory Group for Molecular Screening and Toxicogenomics, or the EMGS committee. And so Jason and I, through this EMGS committee, have been very involved with the OECD's AOP program, the Adverse Outcome Pathway program. And so we're here today to talk about AOPs. We don't want to talk just about what they are and, you know, this is sort of an AOP promotion webex. It's really about why we think they're important for the EMGS, and how we think the EMGS could be involved in advancing the OECD AOP program. So I'm just going to give you a couple of the punchlines that we want to get to in the end and overall before we begin the presentation, so you know where we're taking you and why we're here. So the AOPs are a framework for organizing information to help us understand how chemicals interact with the molecules in cells to lead to adverse outcomes. If you've heard of mode of action, then you know what an AOP is. They are very, very simple concepts. Ever thought about or used the mode of action framework developed from the World Health Organization? Then the AOP framework grows heavily from that as well. Just before I go on, Bob, is it possible to mute everybody while Jason and I are delivering the WebEx?
Yes.
So that's fantastic, Bob. Can you hear me now?
Yes.
Okay, terrific. So yeah, a framework for organizing information, but the difference, and very similar to mode of action, but a major difference is, one of a few important ones, is that AOPs are published in a very novel publishing format. They're published in an AOP wiki, and they're reviewed very differently as well. Publication in the AOP wiki really facilitates use by the research community and the regulatory community. The wiki is searchable. It provides a collaborative tool. AOPs, as they're developed and reviewed, you'll see in the wiki, it's a very transparent process. And really importantly, these AOPs, because they're in wiki format, can be updated in real time. So not like a flat file that you've published in a journal that you have to write a new version of every 5 to 10 years. The wiki format allows us to update AOPs as new information, new tests, new data are available. So these AOPs are becoming increasingly central elements in chemicals risk assessment. And so we feel, Jason and I feel, that it's really important, therefore, to make sure that AOPs in our area and in genetic toxicology are developed accurately and reflect the current knowledge and state of the science. And so today, we're here to explore whether there's interest from the EMGS community in putting together an EMGS EMM review board. So EMM being our journal, Environmental and Molecular Mutagenesis. So the purpose of this review board would be to review AOPs in the area of genetic toxicology as they're submitted to the OECD, and facilitate some sort of complementary publication of the AOP in environmental and molecular mutagenesis. And these publications would be about the AOPs themselves and about their applications. So our intent is to tell you a little bit about AOPs to begin with, and then bring us back to how the EMGS can be involved. So Jason is an international expert in AOPs, has been part of the program since its beginning, since very early on, and he's traveled the world giving AOP training courses. So I thought we would start with a little bit of background from Jason on why AOPs are what they are and why they're important.
Okay, so thanks, Carol. I just want to remind everybody before we go along, if you have any questions and comments, please write them in the comments section of the Go Meeting app, and we'll keep track of them and try to address them later on. So yeah, AOPs. Before we get into what they are and how we can help, I want to tell you a little bit about why AOPs were developed and how they're important. And AOPs were really developed as a tool to help move into this vision of risk assessment in the 21st century. Now, in risk assessment, there's all these new technologies, high throughput technologies, automated technologies. We can measure thousands of different assays all automatically, generate lots of data. And on top of that, we have these high content technologies like genomics, metabolomics, and proteomics. And so we have at our fingertips all these powerful tools for measuring all these molecular, cellular effects and get a great amount of information. And so the idea is that risk assessment can be much more high throughput. All you have to do now is take the chemicals of interest, feed them to your automation process, which conducts all of these assays. You'll get all of these dose response curves. And on top of that, you have your genomics, proteomics, metabolomics. Then you take all that information and you determine if the chemical is safe for human or environmental health. Really great, right? It's unrealistic to think that risk assessors can very easily take all of this information and make regulatory decisions from all of this information coming from all of these different directions. Let's say even if you do identify some assays that are affected by chemicals, identifying how that assay is relevant to human or environmental health is strewn across hundreds, if not thousands and thousands of articles across the literature. So again, it is unrealistic to expect risk assessors to be experts in how all of these different endpoints that we can measure are relevant to human or environmental health. So AOPs are to help address this information overload that risk assessors are facing, the data overload and the publication overload. And so a couple of the primary purposes of AOPs are to help link those things that we measure at all the different levels of biological organization, from cellular to molecular to organ effects, and help make those links to points of regulatory significance. And that's done by efficiently organizing the most relevant experimental evidence that's in the literature and put it in a very easily accessible and easy to interpret framework. In other words, it takes all the complex experimental evidence that's in the literature these days and distills it into context and relevance. We're going to talk a little bit about the documentation process that's involved. There's a very rigorous documentation process and evaluation of this evidence. But ultimately, it makes this stuff very easily accessed. It's machine readable, it's searchable, it's open access, it's linked, so everyone can have access to the information, and it's easily, ultimately, it's the goal to make all of this data much more easily interpreted by non-experts.
Yeah, so we need to begin by talking about what AOPs are. Jason's told you what the vision is here of having this information available to us in an accessible format so that we can easily look across papers and consolidate the information and consolidate data sets together to help us understand exactly what they are. So the first thing that people look at when they start talking about AOPs is usually something we call the flow diagram. And that's what we have here at the top. I don't know if they can see my cursor when I'm pointing, but we have molecular interaction leading through different levels of biological organization at the cellular, the tissue, and the organ response to adverse outcomes at the organism and the population level. So this is called the flow diagram. But really what an AOP is a framework that describes the knowledge that's aligned with this flow diagram about how a toxicant or groups of toxicants would interact with molecules to cause an adverse outcome. And these are relevant to both human or ecological health. So it's really a way to organize all this information. The flow diagrams that we're showing here are based on biological plausibility, highly biological plausible, highly mechanistically understood AOPs. There's one kind of AOP that would have what we'd call a high weight of evidence, but we can also have hypothetical AOPs that are based on other kinds of more sort of statistical inference and other things. At the end of the day, after you have an AOP, what you have is a hypothesis about a mode of action, really, and you have a set of test methods that are described within the AOP that you can use to measure whether a toxicant is addressing this MOA or not.
So one thing I want to stress is that AOPs are not biological pathways. They're not biochemical pathways. They don't describe all of the biology in between all of these steps. It's a framework for organizing evidence.
Mm-hmm, mm-hmm. And that brings me to the building blocks, Jason, because there's two main building blocks. One is the key events. So these are the big boxes that you see in the flow diagram of the AOP. So these are called key events because they're the functional unit of observation that you would test. So the key event shouldn't describe absolutely every single thing that's going on from the start of the interaction with the chemical right through to the adverse outcome. These are the observable changes in biological state that you would measure with some sort of test method. Each of the key events, the blocks here, are essential to the adverse outcome pathway, but not necessarily sufficient in and of themselves. And when you're building an AOP, one of the big parts of the jobs is describing and defining the methods that you would use to observe or measure each of these key events. And then within there, of course, you have to consider things like the taxonomic and sex and life stage applicability. You'll see that the AOPs are bookended by two specialized key events. The molecular initiating event in green on the left is the initial point of interaction of the chemical with molecules in the organism. So this is the thing that begins the domino effect. And the adverse outcomes. And these are endpoints of regulatory significance. So generally, these are an endpoint that's associated with an established protection goal or an accepted regulatory guideline toxicity test in many cases. But I would say that when you look at an AOP, people tend to feel really uncomfortable because, as you mentioned, they look like a really oversimplified description of the biology. But I would say that the meat of the information is in the key event relationships.
Yeah, and the key event relationships, one of the reasons people kind of overlook the key event relationships is because they're often shown as these little tiny arrows in these flow diagrams. So I often, when I give these kind of talks, I like to emphasize that really there's a ton of information in these arrows. They should probably even be bigger than the boxes. They represent the functional unit of inference of the AOP. They contain all of the evidence that defines the relationship between these things that we measure. In each of these arrows is all of the documentation of and evaluation of the evidence that shows that when one of the upstream events is perturbed, that there's a following effect in the downstream event that can be measured. And we'll talk a little bit about the types of evidence that go into there, and there's also some degree of quantitative understanding about these key events.
I would say, Jason, that if you look in the key event relationships, they're generally quite substantially longer and there's more meat in them than the key events, and they're really the toughest part to build. It's really where you're defining, when we say biological plausibility, that's where we're defining exactly the mechanisms that we understand and are published that link one event to the next.
A lot of the complexity that people feel is missing from AOPs is found in these little arrows. They're shown as little arrows, but they contain a lot of information. And so they have a lot of the evidence. And there's different types of evidence that add to this evaluation. Some of the most important types of evidence are biological plausibility. Biological plausibility is a description of our current understanding of the biology behind these relationships. And in many cases, these types of relationships have been studied for years and decades. So this point alone contains a lot of information in itself. Another important point of evidence that's included is that the different key events are essential to the adverse outcome. And there's different types of experimental evidence to support that. And then Carol talked about some types of evidence that are more data-driven, and this is the empirical support. And these are experimental evidence that demonstrate a dose-response relationship, or a temporal relationship, or an incidence or frequency relationship. And of course, throughout all of this process, we have to very clearly document all the uncertainties and inconsistencies. And there's some of these types of evidence add more strength to the others. For example, something that's been studied for very long and has a high biological plausibility lends very strongly to the weighted evidence evaluation. And so importantly, all of these types of evidence in the knowledge base or in the wiki come with this weighted evidence evaluation. And so every time you present a relationship, you have to say if there's a lot of data supporting evidence, and if there's not, then the weighted evidence call is weak. But if it's an area that's been studied very thoroughly and has high quality studies with corroborating studies, that the weighted evidence is very high. So the key event relationships just have a ton of information in.
Yeah, that's a good summary of that part. I would say that building that weight of evidence evaluation is really one of the most challenging parts of building the AOP. That and getting agreement on the flow diagram. So that was pretty tricky for us to do initially. I guess then quick recap, because now we've talked about what AOPs are, and now everybody knows what they are, and then we'll move on to how we might use them and what's being done in genetic toxicology. So to recap then, we have key events. These include a molecular initiating event, key events along the chain, as well as an adverse outcome. That's the specialized key event that we see at the end. And these are all things that we can measure. And then we also have the key event relationship, which is the evidence that links an upstream key event to a downstream key event. And then finally, when we put these things all together, we have the flow diagram that describes the entire AOP. And it's really on the front page of the AOP wiki where you see the full description of the AOP. Because each of these elements within the AOP, as you'll see when we move forward, these are modular and other people will reuse them. So the only thing that's unique to the AOP is the actual description of the overall AOP.
So yeah, so now that we've described kind of the structure of the AOP, there's a couple really important concepts, and we've touched on these things a little bit already, but we just want to drive them home because we feel they're really important. So again, one of the most common misconceptions with AOPs is that they're oversimplifications of biology, and that they're intentionally designed that way. They're meant to be a pragmatic simplification of all of the biology. They represent only the things that we measure and the evidence linking those measurements along the way. A way to think about, one of the reasons that they are simplified is because it really makes their development a lot more streamlined. If you had to describe all of the biology along the way, these things would be massive and it would take forever to develop.
They would be very useful for risk assessment purposes.
And so making them simple linear units is pragmatic for their development and review. And so a good way to think about it, a good analogy, is that a single AOP represents one way to get from A to B, but it doesn't represent all ways. And so just to make things more easy to describe and review. But
you know, I would say, just to give to orient people as to the level of detail in an AOP. So I would say it's about the equivalent of a review paper, of a detailed review paper.
I think it's more than a review paper.
Do you? Yeah. I guess I wouldn't disagree with that, actually. There's quite a lot of information in these AOPs, so don't be misled by looking only at the flow diagram, basically.
So with that being said, so that leads into the second point, and to continue the analogy of the route on the map. If AOPs describe a single path, well, it's the full map or the full network of all of the AOPs in the knowledge base that are used to help understand how chemicals can lead to adverse outcomes. The full knowledge base or the full AOP network represents all possible adverse outcomes and all of the ways that we can get there and all of the things that we can measure to help determine if this path is being followed or not.
Mm-hmm. And we can see things like pleiotropic effects of chemicals by merging these AOPs, and we can identify other potential molecular initiating events that might lead, it can converge on our pathway, help us understand mixtures and other things. So that the network is a really powerful tool for sure.
And so this leads to the third important concept, is that AOPs are modular. In order for these networks to occur, they have to have shared components. And when you're developing an AOP, there's a lot of overlap in the information between the different AOPs. And so there's no sense in having authors describe that same information independently of each other. So AOPs are built in a modular way so that the different evidence and measured units and the links between them can be borrowed between AOPs. So in this little example here, there's the animation. Oh, I blew it. But you can borrow the different things, and it's when you share different key events and key event relationships. This is where the networking occurs, mm-hmm. and allows the simple AOPs to become a much more powerful tool in their full completeness of the network.
Mm-hmm. And I think, Jason, this is also why we're new to this game. Basically, the program is not an old program, but these first initial key events and key event relationships, we want them to be of the highest quality and really reflect the state of the science, because others will come in behind us and reuse these elements. And so we want them to be the best that they can possibly be. And that kind of aligns really with the fourth important concept, which is that AOPs are living documents. I mentioned this right at the beginning, but for those of you joining late, AOPs are published in a very novel format. They're published in an AOP wiki within the AOP knowledge base. So everything is stored there, and through publishing on this format, it facilitates collaboration and it facilitates crowdsourcing. Others can go in and they can build on the work that you've done. A really good thing that it does is avoids duplicative effort. If I have built a key event or a key event relationship, there's no need for others to do the same. They can easily find it because the AOP wiki is searchable and accessible. They can easily find it. They can build on it in whatever way they want to. So finally, a critical element is that these AOPs, because they're in a wiki platform, can be updated at any time. So you produce a new methodology, and you're now able to go into the wiki and align your new methodology with different key events and feed into AOPs. You produce a new data set that builds on the weight of evidence supporting an AOP, and you can plug those data in immediately into the AOP wiki.
So we've talked a lot about what AOPs are and a little bit about what they're not, and a few of the important concepts behind the AOPs, but maybe some practical examples. I'll talk about like an ecotox example, but then Carol's going to get into some more EMGS relevant examples. Mm-hmm. So a simple example here is, imagine you're a risk assessor in the 21st century, and you're
that's me over there on the right.
You're not a risk assessor.
Regulatory scientist of some sort.
And you've been tasked at assessing a particular chemical or a group of chemicals, and you've sent them over to the ToxCast program, and you get your results back, and you find that a couple dozen assays are positive in the ToxCast battery of tests. And you can see that different assays have different sensitivity, and in this particular example, the most sensitive assay is one for thyroperoxidase activation. And as a risk assessor, trying to interpret ToxCast results, it's not immediately obvious about what the relevance of this change in an in vitro test actually means for human and ecological health. Mm-hmm. And so what AOPs do is they allow a non-expert to access the years and years of research that have gone into this particular endpoint and find all the most relevant evidence linking that change to something of regulatory significance in a structured and easy to understand way. So here in this example, we see an AOP network showing that thyroid peroxidase inhibition is linked to some human and ecological adverse effects. So what that allows a risk assessor to do is to quickly access this information. And the long goal, I don't think we can do it yet, is to have quantitative linkages where we can actually quantitatively understand how much TPO inhibition is required to reduce to an ecological effect, such as metamorphosis impairment. But right now, AOPs are more in a qualitative state, but that's still very useful. This allows a risk assessor to come up with a hypothesis that TPO inhibition might be related to reduced survival of wildlife, and provides some well-documented relationships to things that you can measure to test that hypothesis. And so the relevance of this molecular change in an automated in vitro test is much more apparent to a non-expert.
I like the so relevant. So, I mean, I guess the long-term objective, Jason, would be to have AOPs addressing all of these different potential molecular initiating and key events here, and be able to, you know, really have some sort of predictive toxicology that allows you to quantify how much of this you need to get before you would get to the adverse outcome. But one of the things I also liked about the slide that you showed was you're interested in ecotoxicology and I'm interested in human health, and you could see how the information and the data and the test methods across these different areas of toxicology can converge in AOPs and really help to inform each other.
Yeah, that's exactly right. So it allows a lot of collaboration between non-related fields as well. And so this is one simple example that we've highlighted. And but there's so many other potential applications for the use of AOPs in regulatory context, but also in a research context. One of the most important ones, we think, is the use of new approach methodologies in integrated approaches to testing and assessment.
So I just want to pause there, because I think this is a really important point in the conversation to stop and just say, what is the OECD and why is it important here?
So the OECD, for those of you that are unfamiliar with the OECD, it's the Organization for Economic Cooperation and Development. And the OECD's primary interest is to ensure economic well-being for all of its member countries. There's like 30 or 50 countries.
Yeah, 30, 35.
Don't quote us on that. But and so at first, it might not be obvious what toxicological testing or genetic toxicology testing has to do with economic well-being. But so you can appreciate that chemical testing is extremely important to world trade. All of the products that we buy and sell and trade around the world have chemicals in them. And in order for this trade to occur, we all have to agree which chemicals are safe. And in order to agree which chemicals are safe, we all have to agree on the methods that are used to test those chemicals. And so the OECD is very interested in harmonizing chemical testing to facilitate chemical and basically all of trade in all of its member countries. So it's very, very invested in toxicology testing.
So the test guidelines program is really important in chemical testing, of course, because if you live in France and you run a test following the OECD's test guideline, then I can use the data in Canada. But people will tell you, if George Douglas is listening in on the call, I'm not sure how long it takes to develop one of these test guidelines for these conventional regulatory tests.
Exactly, yeah. So the test guideline program, well, it's extremely powerful and useful. It's very, very time-consuming. It takes some time. And so the idea between the integrated approaches to testing and assessment, it allows the flexibility to use novel methods when sufficient evidence is provided for their relevance. And so the AOP program framework blends perfectly with this goal. It provides all of the evidence and very rigorous documentation of the evidence behind that evidence, or the evidence behind that evidence.
That's a good one.
Yeah.
Carol and Brian, there was a question from Daniel Roberts. It's relevant to this. So the question is, a general 100-foot view comment. If OECD is an AOP review board, would an EMGS review panel for Genotox AOPs be redundant or duplication of effort? Would OECD recognize this specialized review board?
That is an excellent question, and we are going to address that shortly. It's exactly where we're going.
Stay tuned for our last 10 slides.
Yeah.
Thanks for that. Yeah, we'll get there.
Okay, so basically, I hope we've described the relevance of the OECD, or why they're so involved in this and invested in this. And so the IATA is probably one of the most powerful approaches to integrate novel testing methods, such as those that are developed by EMGS and its members, into regulatory relevant testing paradigms. And then there's a whole bunch of other potential applications that are listed here. One that I'd like to also highlight is that as a collaborative research tool, just when you organize your the knowledge in your field of research in this way, it really identifies data gaps. And so it really helps you as a researcher with your experimental design, where you can design experiments that will make the most impact and help get your science to the risk assessment.
And testing gaps as well. And I also think that when you start thinking about AOPs in the context of what experiments you're doing, it really helps strengthen your experimental design of the work that you're conducting in your lab. So yeah, very important tool for those kinds of applications as well.
So I described like an ecotox simple example, and then we have a few other like potential applications. But I think maybe this audience would be much more interested in a DNA damage EMGS relevant AOP.
Well, so you know, EMGS has been thinking about AOPs for quite a long time. We've had 2 workshops on AOPs, and we've had symposia and other things on AOPs. So I'm just going to give you a little bit of the history about how the EMGS has been involved to date through a description of a case study in mutagenesis, and that's alkylation of DNA leading to heritable mutations. So back in, I'm going to say 2013, we had our first workshop on AOPs, and we brought together a small group to say, you know, what should we, here's this AOP program, do you guys think it's important? If you do, what should be the first AOP that we develop? And the group decided that alkylation of DNA leading to heritable mutations would be a good place to start. And maybe this seems like a bit of a strange place to start because it's not really used a lot in regulatory decision making, but it's one of the best characterized modes of action in genetic toxicology, and we understand it very well. And so we thought it would be a great place to start for that reason. The plan was to look at this AOP and figure out how the AOP program could help us to provide context of use for the new methods or technologies that were being developed, how we could use information from somatic cell mutations to inform germ cell mutations, and could we, even through the development of this AOP in this area that was really, you know, sort of the roots of the EMGS, one of the first areas of study in genetic toxicology, could we still identify testing and data gaps in this field? And of course, our group at Health Canada is very interested in increasing regulatory use of germ cell testing, because we know that germ cell mutations contribute to a huge number of human genetic diseases. So we had that sort of in the back of our minds too as we were moving forward with this. So just let me walk you through this AOP and how it came to be. So the first thing that you need to think about is your molecular initiating event. So we thought about what kinds of chemicals that we knew about were alkylating agents. We know these chemicals can add ethyl methyl propyl groups to different places in DNA. And so we've consolidated all that information. When we had the information that we could use to build the AOP, we saw some information for a variety of alkylating agents, but really the database for this AOP was heavily biased towards ENU.
And so I just want to interrupt for one moment here, because this is where another common misconception occurs. One of the important points that Carol made earlier on in the presentation is that AOPs are actually chemical agnostic. They're supposed to be describing how any chemical can lead to an adverse outcome. But of course, the information that informs the AOP comes from specific chemicals. So here, this AOP is heavily dependent on ENU data, but is theoretically relevant to any chemical that can alkylate DNA.
Because it's the same type of alkylation event. That's right. So chemicals can alkylate DNA. And we know the next thing that happens after a chemical alkylates DNA is that, quite generally, those alkylatics are readily repaired. So in the AOP, we describe the primary repair protein, which is alpha-guanine transferase, AGT. And we describe how AGT irreversibly binds to the alkyl group and is thus inactivated. It's a suicide enzyme. And so for that reason, alkylating events induced by ENU are very efficiently repaired at low doses, but when AGT is overwhelmed at the high doses, those alkylatics are retained. And we also know AGT is great at specific types of addicts, but not all types of addicts. So let me just go on here to the selection of the measurable endpoints. So we had to think about, if we were thinking about alkylation of DNA and leading to heritable mutations, which events along that pathway would we actually measure for regulatory purposes? So up the top here where there's a green check mark, it means these are things that we could and do measure. So we can measure the presence of adducts in DNA. We can measure whether they are repaired or they're not repaired, that these adducts are retained. This was caused quite a lot of conversation and development of this particular AOP, and that's the fact that we know that replication is essential to the creation of mutation. However, we don't measure mutation. We don't measure replication. So that's why there's an X here. This step in the pathway of replication has to be very well described in the key event relationship, but we're not measuring it, so you won't see this in our flow diagram. We can measure after replication whether these mutations have occurred in somatic cells or in sperm in this case. We're not very good at measuring mutations in females yet. Which we don't measure, but we can measure offspring with mutations. And this, of course, is the big challenge in germ cell genetic toxicology. It's the lack of really efficient tools to measure offspring with mutations. So we'd like to be able to quantify this AOP to reduce the needs to ever measure mutations in the actual offspring. So this leads us to our flow diagram, which is described here and found. You can see at the bottom, if you click that link, it would take you directly to the AOP wiki. So we have alkylation of DNA as our molecular initiating event. KER1 stands for key event relationship number 1, leading to, we called it insufficient or incorrect DNA repair as KE1. There is a lot of information about what pathways are involved in DNA repair in this key event. And in fact, this key event is the one that's evolving the most now as the community builds on this AOP, and this key event has now been renamed. So if you click on the wiki, you'll see that this has a modified name now.
Hey, Carol,
So yeah?
there's a question related to the last slide. Why is replication not measured?
We make sure that replication occurs, but we don't measure it to it's
regulatory testing.
Yeah, it's not part of the regulatory testing battery. It's that we make sure that it has occurred. It's essential to ensuring that the mutation is created as a result of the adduct, but it's not something we measure. So we have a key event relationship here leading to mutations. If these mutations occur in male germ cells, then we can have inherited mutations. So these mutations are in the offspring, and it's increased number of mutations that you would be able to measure in all the tissues of that offspring. Just
to go back to that question, that the replication would get captured in the key event relationship under something like biological plausibility.
Yes.
It describes the relationship, and it'll describe that replication is absolutely required, but it's not something that's regularly tested.
It's also described in the methodologies because the experimental design requires you to set up an experiment to make sure that sufficient time has passed, that you ensure that replication has occurred. So it's a great question.
Yeah.
And that's where it is mentioned in the AOP.
And it actually rolls quite nicely into this this next slide, where we're talking about the different types of evidence that are used to support this AOP. We mentioned them before. There's empirical evidence. You can have dose concordance, temporal concordance, and incidence concordance. But often experiments in the literature don't usually have the type of experimental design that is ideal for supporting these relationships. So it's kind of one of the challenges in developing an AOP is finding the right experiments to support these relationships.
And perhaps this isn't surprising, but it was a little surprising to me. The most challenging part of building this initial AOP was finding the empirical evidence to support the AOP, because studies don't necessarily measure more than one key event in a study experiment. So we had to reach across studies. Moving forward, what we're trying to do in my lab is to build multiple key events into experimental design so that we're right from the start providing evidence supporting AOPs. But these are some of the types of empirical evidence that, or these are the types of empirical evidence that we consider. So one is dose concordance. This is a fantastic paper by Swinburne et al. In 2008, which is a great demonstration of dose, and it could be also converted into incidence concordance. So in this plot, you'll see adducts are displayed with diamonds, and mutations are shown in the open circles. So you can see there are, we begin to see adducts right off the bat following an MMS exposure, but it's not until we surpass a certain concentration that we begin to overwhelm.
You were about to say threshold.
I wasn't going to say threshold. It's a break point. It's a nonlinear. So it's not until we overcome the AGT that we begin to see these increases in mutation frequency.
And so this provides excellent an example of how adducts occur earlier in dose than mutations. So perfect example of dose concordance.
And then the next piece, of course, is temporal concordance. And here we really had to look across studies. In this example, this is a paper by Saylor et al. 1997 showing that in male germ cells, we see adducts, within hours of an exposure to ENU or DEN, whereas Jason, I believe these are your paper. This is your paper with Francesco and I. Maybe you should jump in.
So this is a good example of how AOPs can help you design experiments to support the regulatory relevance. So to our surprise, there wasn't a lot of temporal evidence linking adducts to mutations. And so this experiment was designed specifically to help support that. And it's basically showing that mutations from ENU don't occur in sperm immediately after the exposure, and that they tend to peak about 70 days after the exposure. So what this really shows is that in time, adducts occur at an earlier time point compared to mutations.
And then the final piece of evidence was putting it all together for incidence concordance. And here we had to look across, this figure that I'm showing here probably had 10 or 12 studies worth of data in it. And it shows in the blue diamonds here, the incidence of alkyls per dose of alkyl adducts per dose of ENU. And you can see there's more alkyl adducts than there are mutations in either sperm in the red boxes here or in the offspring, which are the triangles here. And that these data basically demonstrate that there's more adducts than there are mutations in sperm. And there's an approximately equal number of mutations in sperm as in the offspring from these experiments, specific locus test and LACZ mutation assay. And then finally, we're looking for evidence of essentiality. So if you block one of the KEs or you enhance one of the KEs, what happens downstream of that to the subsequent key events or the adverse outcome? So in this example from Lei et al., we have evidence supporting that overcoming DNA repair is required for mutation to occur, and we have this through. This is actually a line of mouse that has a AGT overexpression. So here we have exposure to 80 milligrams per kilogram of, I believe it was ENU. I'm not sure, I can't remember now, but an alkylating agent, and we see this very significant increase in the numbers of mutations following the exposure. And when AGT is overexpressed, we see these coming down. So overexpression of inadequate repair basically leads to the predicted downstream impact on the mutation.
So this is a good example of how chemical-specific data is used to support a generalized key event, the inadequate repair key event.
So how does this look in the wiki? You can go into Google and you can type AOP wiki, and it should be the first thing that comes up. And when you go into that link, you'll see across the black bar at the top, there's a bunch of searchable elements. So click on AOP and type in alkylation of DNA, and the first AOP that pops up will be alkylation of DNA in male premeiotic germ cells. I'm very sure it's the first one. Don't look at the second one, look at the first one. Alkylation of DNA in male premeiotic germ cells leading to heritable mutation. And when you click on that title, that'll take you on to the front page of the AOP. The front page of the AOP is the only thing that's specific to the AOP. Everything else is modular. So this is where we get into the specific details about the overall pathway and even its regulatory significance. So I'm not expecting you to read this, but you'll see there's some tables here. So on the front page, when you go through these tables, you'll see hot links. Here you can click on these hot links and it takes you to the descriptions of each of the key events. And in the second table below it, you can click on these hot links and they'll take you to the description of the key event relationship. And in the bottom here, this sort of network diagram is something that's created by the wiki itself. So it's showing my AOP from the blue boxes, the molecular initiating event, through the key events to the adverse outcome in yellow. But these other triangles are showing me other AOPs that are now connected to my AOP. So what's being developed that's reusing elements of my AOP. So very quickly, you can go to an AOP that you're interested in. You could see maybe what other key events are downstream effects that that molecular initiating event may initiate. And then once you've clicked on these hot links, just to show you here on the left side is a key event description. So some of the text that you might see in describing the key event here, you'll see some text in black and some text in blue. So one of my colleagues, Dr. Vinita Chauhan, is reusing our AOP elements to build an AOP associated with deposition of energy, so radiation exposures. And she's added new text to this in blue, so you can quickly see how these can evolve and develop over time. And the same goes for the key event relationship over here. And this has a ton of information in it, so I'm just really giving you a little bit of a snapshot here. So the status of this AOP. This was one of the first AOPs ever endorsed by the OECD, by the national coordinators and the Working Party on Hazard Assessment. And so we had a very productive review process. And I know a large variety of EMGS members were actually on. I think there were 7 reviewers in total for my AOP, and some of you I think are on the line today. And it was a very good process with an open dialogue, and so now that AOP has been reviewed and accepted by the OECD. It's published also in the OECD's publication series on adverse outcome pathways, but we also published a report on this in environmental molecular mutagenesis. So the first Genotox AOP was published in EMM, and so we're sort of coming back to this full circle. And so now, as I was mentioning, Vinita borrowing some of my elements, but there's other work going on, and this is an example of some things that we're thinking about now. So across the top is our alkylation of DNA leading to inherited mutations, and one of the things we're developing now is an AOP that leads through alkylation of DNA through to declines in sperm count and infertility. And we can also build upstream of that. So there's more than just alkylation of DNA that can lead to mutations. Of course, there's all kinds of other molecular initiating events. In 2017, the Health and Environmental Sciences Institute's Genetic Toxicology Technical Committee became very involved. And a workshop was put together that was joint with the Genetic Toxicology Association in 2017 that was led by Drs. Jennifer Sasaki and Mike Schuler to ask questions of the gene tox community about, how we can use the AOP framework to inform mode of action and for risk characterization. So it's a very productive workshop, lots of fantastic discussion. And in the end, it was decided that this was a great framework for developing regulatory testing paradigms and for characterizing mode of action. And so the workshop encouraged the development of AOPs that end in permanent genomic damage. Now this paper, I'm really pleased to say, was published recently in Environmental Molecular Mutagenesis, keeping with our theme, but it's in the 50th anniversary special issue as one of the sort of forward-looking, the future forward focus of the EMGS. So you can now go, that art issue is open access, so it's freely available to everybody. So spread the word, and you can see what the gene tox community is doing in this area now, and what their thoughts are. But some working groups emerged from this. One of the leaders is Stefan Buehler, and one of the things that we've been doing is working with members of the AOP working group within the GTTC on this particular AOP, which is the majority of the work is being done by Narcho, a PhD student in my lab. And the idea is quantifying how much oxidative DNA damage you need before you begin to see mutations. And so in this AOP, we have oxidative DNA damage. You can see now how the inadequate repair key event now, instead of the insufficient or incorrect DNA repair, so you can see how these key event names are evolving, can lead through DNA strand breaks, again, through inadequate repair to either mutations or chromosomal aberrations, or directly through inadequate repair to mutations. And so here we're aligning some of the most novel cutting edge methodologies that we can use to describe each of these key events to help us put together new testing paradigms. And this network is continuing to grow. Now we're not the only working group within HES-CG-TTC that's building AOPs. So you can see across the top here is an AOP that begins with binding to DNA topo 2 cleavage complex and disruption of replication forks, leading to these different kinds of adverse outcomes. We've got binding to DNA polymerases and disruption of DNTP pools. You can see here the Venita's AOP, which is direct deposition of energy. This AOP is focused on primarily on radon and its ability to cause lung cancer. And in parallel, there's a growing network of aneuploidy AOPs with involvement from Francesco Marchetti and Mike Schuler and others. Francesco's AOP is actually just about to be launched into the external review process. So the state of the science here is, the state of the review process here is Venita's AOP on radiation exposure is about to go into the review process, as is Francesco's. And the oxidative DNA damage AOP has just been submitted for an internal review that Jason will discuss in a few minutes before it can go to external review. But there's a whole bunch of, in addition to publishing these AOPs, so much work goes into developing them and discussing all of this, and there's a variety of applications. So there's all kinds of different papers emerging from this work, not just put an AOP in the AOP wiki. And so this is an example of reports that came out of an international workshop on genotoxicity testing that focused on the development of NUgenicity-associated AOPs and their involvement in carcinogenicity and heritable genetic effects.
So as I hope Carol has convinced you that the AOP development community in the genetic toxicology field is very healthy. There's a lot
very active.
There's a lot of people contributing to AOPs. And there's a lot of publications going on. But ultimately, when you're developing an AOP, what you really want is to get that OECD stamp of approval. Mm-hmm. So the OECD, we talked a little bit earlier about why they're important and why they're so invested in this, but they've formalized this whole process of the development and the review, the endorsement, and the publication. A lot of this information is made available in guidance documents. Probably the most useful document is the user handbook. This is very, very specific guidance on how to actually develop AOPs and how to provide the weight of evidence that's required to support the relationships. And of course, all of this information is stored in the knowledge base, which is primarily accessed through the wiki. Now, I want to describe for you a little bit this endorsement process. We already talked about at length quite a bit about how AOPs are developed. So you make your modular key events and key event relationships, and then you assemble them into AOPs, and then within that process you evaluate all of the evidence, and all of this is done collaboratively over the wiki. But what we haven't really talked about much yet is this review process. The review process is kind of a two-step process. There's first an internal review, and this is a compliance check, and this is done by an AOP expert in the OECD, and this is just done before you go through the lengthy external review process, just to make sure that your AOP structurally follows all of the rules that Carol and I had described earlier. And this is just so that you don't go through all of that effort just to find that there's major problems and then have to start all over again. So once that's done, there's an external review process. And this is done by an external panel, much like a regular review, but this is not a typical journal review. There's quite a few major differences. The review is overseen by a review manager, and this review manager actually facilitates an open dialogue between the authors and the reviewers. The authors and the reviewers are, their identities are known to each other, and they meet by teleconference and discuss some of the issues that come up, like describing the flow diagram and which things you can measure and which things you can't, and so on. And so there's lots of open dialogue. There's usually at least one, if not more, round of revision. Again, all open dialogue. This is all facilitated through the wiki, so you can track all your changes and edits, and all of the comments and changes are documented and made publicly available in the wiki as well. Mm-hmm. So this is quite different than a standard journal review.
Mm-hmm. The review manager is sort of like a handling editor, really. The role that they serve, they're consolidating the comments from the reviewers and helping to facilitate with the author how the AOP really should be revised.
Yeah. And then, so once you go through that pretty rigorous review process, then the AOP is forwarded to a different branch of the OECD for endorsement. This is endorsed by two major groups, the Working Group of the National Coordinators for the Testing Guidelines and the Working Party on Hazard Assessment. And these are the major risk assessment groups of the OECD toxicology section. Mm-hmm. And once you get these stamps of approval from the OECD, this is what gives your AOP that high confidence to be used for regulatory applications. Mm-hmm. And then after you get this stamp, the AOP is published. And again, we talked about how publication for AOPs is quite different than a standard publication. There is a static PDF version that gets published as part of the OECD AOP series, and you can download these PDFs and you can cite them and so on. But the version that exists on the wiki is a living document, and it's open for further modification. So not your standard publication.
Mm-hmm. No, definitely different from the normal kind of publication. And I don't think that EMM would want to publish the static version of the AOP. I mean, these are absolutely massive documents.
So this is kind of where we come to that question that was asked to us earlier. Mm-hmm. Why, if this process is already being done by the OECD, why would EMGS want to be involved in this? And this is because, you know, the genetic toxicology community is not the only ones making AOPs. There's a lot of buy-in in the toxicology community at large, and so the AOP program has grown larger than the OECD can handle itself, and there's a major bottleneck in the review process. So much so that many of the AOPs that are relevant to the EMGS are kind of stuck at this review process, and so there is a low representation of EMGS relevant AOPs in the endorsed and published AOP lists.
And I think actually the timing is really right here. So this panel on the right-hand side that you've shown actually reflects what's going on right now, because I do think there's only one Genotox AOP so far, and that's the one we just discussed. And so we want to make sure that we're in early, so that these early AOPs are built on the best possible information.
So the OECD has recognized this bottleneck, and so they're kind of exploring different options. And one of the most promising ones is they want to outsource the review of AOPs. And to help with this, they've developed a standard operating procedure for this whole review process that we described. And they've explored many different partnerships, but one of the most promising ones and probably most exciting ones for us is that they want to collaborate with scientific societies, specifically ones that have journals facilitated with them, that have this machinery for review already with some infrastructure. And so they are willing to allow the AOPs to get reviewed by third parties as long as they agree to follow this review procedure that they've set up. And then if they do follow this, then these AOPs will be suitable for OECD endorsement, which is the stamp that the risk assessors wanted to have.
Yeah. So, of course, this brings us to the opportunity for the EMGS and for EMM to come together and to see if we want to give back to the community at large to help advance this process and to make sure that things are done right, to inform how regulatory paradigms can be built and how risk assessment applications will be using the highest quality AOPs. So our discussion is about whether the EMGS is interested in creating a joint EMGS EMM, essentially a review board or a mini editorial and review board on genotoxicity-associated AOPs. So in this committee that we form, or review board that we form, EMGS members would serve as AOP either handling editors or reviewers for AOPs in the gene tox field. Really happy that we have Jason O'Brien here today, so a long-standing EMGS member and an expert in AOPs who's willing to take on the leadership role in terms of being the OECD point of contact and helping the society move forward. Our review of an AOP, we would have a template that the authors would fill in that aligns well with the overall AOP summary report that you see in the AOP wiki. And the reason for, we want to keep this relatively short and compact, this publication. We don't want it to be the entire AOP, but we want it to highlight the key features of the AOP at the time at which it was published and its potential applications. And so this is something that we would need to think about. What exactly would the publication look like? What would the template look like? But AOPs in and of themselves are only one piece of this, because there's a variety, as we mentioned, of applications for AOPs. So this editorial board or review team would also handle other AOP-associated publications for EMM. So for example, publications that relate to test paradigm development that have used an AOP, applications of the AOPs in risk assessment in different kinds of case studies, the development of modes of action analyses that are applying AOPs, or building that quantitative, the holy grail we talked about, the quantitative association between the molecular tuning and then the adverse outcomes, so those sort of predictive tools. These would all potentially comprise parts of this sort of mini section that we would have in the AOP process.
And along with that, clearly one of the major benefits would be providing a parallel stream to prioritize EMGS relevant AOPs, so that we make sure that the EMGS has vouched for these genetic toxicology AOPs and that they're of the highest quality and can be shared and agreed upon, and get these stamps of endorsement. So this will really help facilitate the development of IATA type testing paradigms.
Yeah, we are the experts, so we should really be the ones doing this. And so then following our review, we would have the AOPs going back to the OECD for their final endorsement. So where are we now? Just before we open everything up for discussion, we have had the opportunity to discuss this idea with the EMGS Executive Board and a little bit with Council pretty quickly in a very small amount of time at the face-to-face meeting. We've also had discussions with EMM, Bhaskar Golapudi, the editor-in-chief, and the Wiley representatives, and there's quite broad support in moving forward to at least see what members think. So at this stage, we're reaching out to our membership to know what you think, how you think the EMGS should or could be involved. So finally, I guess the bottom line is we're asking, are you interested in this? Are you interested in EMGS collaborating with EMM to implement an AOP review and publication process, to review AOPs before they're endorsed by the OECD, and also to serve as review specialists for associated publications. We're asking if we'd like to have a publication series on AOPs within EMM. We have to think about whether we're happy with adopting the OECD's AOP review process. The major difference is that it's open and transparent and that there's this dialogue. Other than that, it's a pretty standard review. There's some special questions relating to AOPs, but the review process is quite similar to a journal review.
The SOP that they've developed has a lot of flexibility in it as well. But yeah, there are some major differences.
Yeah. And we think it provides the opportunity for the EMGS gene tox experts to bring high quality AOPs to the regulatory community. So we are going to follow up with a survey to ask if people are willing to contribute their time to toward this initiative. But at this point, we have time for a discussion. So Bob, I don't know if more comments or questions have come in via the comments boxes. I know that if we open up the lines widely, sometimes there's so much background noise that we can't really hear anything. So it would be really great if people have questions, if they could type them in the comments box and Bob could read them out.
Yes, thank you. So there are a couple of questions and a couple of comments. Bill asks, how does OECD endorsement sit with the idea of AOPs as a living document?
So when the OECD national coordinators set their endorsement, it's marked at the time, it's given a timestamp basically. This is when it was endorsed. It's very clear what the state of the AOP was when it was endorsed. Beyond that, the OECD, I don't think, has made any sort of decisions about whether they'd go back and, when these AOPs evolve to a significant extent, how they're going to update their endorsement. So it's a very good question.
Yeah, so the AOP program itself is quite new. And so we're only, one of Carol's AOPs is one of the few that have actually been through significant revision. And so we haven't had to really address that to any major degree. So there's ongoing discussion on how to address that. But what the endorsement really is, it's not so much an endorsement of the content in there, it's more of an endorsement of the process that was taken to establish the AOP. So the OECD is basically saying that we recognize the amount of rigor that went into evaluating the evidence in this set of relationships, and we stand by the process. And at this time point, we agree with how that process was executed. I don't think that they necessarily endorsed the specific content, so I think that was the kind of loophole to.
That was being the guinea pig in the process. We had a lot of discussions about that, yeah, but they have endorsed the process, you're right. And when people update or add information, it's very clearly noted what has been added and when, so you can see in a transparent way what's been modified. And so it's up to the author, really. They can go back in and they can remove stuff that people haven't have added that they're not in agreement with. But it's the timestamp and it's the endorsed process that it was fully reviewed by an expert community.
Okay, another question comes from Sandy. Lasha Limited currently creating a new program called Captus, a knowledge management system for AOP. Have you reached out to them, as it seems this might be redundant to the AOP wiki?
No, I have not. Is that from Sandy, you said? Because we would like to know more about that, and it would be great if she could send us that information.
Okay.
I don't know if she's still, I think it was Sandy, you said.
Yes, Sandy Weiner. Rosie has a couple of comments. It's high time that EMGS participated in such activities that organize its unique expertise in a positive manner. It also enhances EMGS's reputation. Members already do this under other hats. We would have a collective impact that is unparalleled. Also, I think our expertise would facilitate the creation of better, more realistic tools, and anybody can create a few boxes with obvious event names. It's important to address actual utility of the AOPs for the regulatory community.
Fantastic. Thank you very much, Rosie, for those comments. I did at one point have a pros and cons slide that I didn't include, and I think you've really nicely captured with those comments some of the pros that I had listed. I feel like, many of our members definitely are, she said you're welcome. Many of our members are already involved. We know we have the expertise, and I feel like we should be giving back and making sure that these are done correctly.
What's an excellent example of creating tools that have actual utility is, for example, the oxidative stress AOP that Carol's group is working on, who are deliberately choosing key events that have known novel test methods associated with them. And the flexibility of the AOP framework is that when you develop new tools that have regulatory relevance, you can integrate it into the knowledge base so that the relevance to risk assessors and adverse outcomes is apparent. And you don't have to recreate every pathway all the time. You can kind of just jump in at points where your tool measures, and all that evidence is still relevant.
So thank you for that comment. I would mention, because it was an open review process, Rosie was one of the initial reviewers for my AOP. So if you're interested in finding out what that was like for her, I'm sure she'd be happy to. Well, I'm speaking for you, Rosie, but you can ask her what that process was like.
So we have another question. Should there be a standard set of requirements specific to the type of study, that is in vitro and in vitro study, that individual studies need to meet to be used as empirical evidence?
Well, when you're including studies to be used as empirical evidence, you should be making statements regarding the quality of those studies. But as of right now, there is no set of standards. Some of the test methods that you use in here are OECD test guidelines. Some have undergone some types of validation, but some are brand new test methods. It's really about being able to figure out how to use information from different sources, different test methods.
That being said, the weight of evidence calls are dependent on that sort of information. So when you have multiple corroborating studies that show the same evidence, regardless of how they were conducted, it adds a lot more weight of evidence than single studies.
But I guess the question is really, should there be a standard set of requirements for studies that are used in that? And I think because the test methods are evolving, that maybe a standard set of requirements isn't the way to go. And it also depends on the type of AOP that you're developing. So we talked about, you can have a hypothetical AOP with very weak weight of evidence, and you might use that information, for example, in a preliminary screen to say, oh, there is some biological activity from this chemical. And so that would be a very different application than a highly developed quantitative AOP with a lot of strong empirical support that you could actually use in a risk assessment. So there's this spectrum of stages of development of AOPs and they have different purposes to fill in risk assessment context. So
in a way, the guidance documentation, it doesn't standardize the types of tests, but it kind of standardizes, or at least recommends, the types of data that can help. So for example, the dose response concordance evidence has to link two key events together over dose. So the guidance documentation helps, but doesn't standardize. It's flexible enough.
Flexible, yeah.
Okay, we have one more comment from Carol. Sandy's comment on the work Lasha Limited is doing. I'm from Lasha and can arrange for someone to get in touch about the work we're doing and opportunities for collaboration and coordination.
Oh, terrific. Thank you, Carol. Carol says thanks to Carol. Are there any other comments or questions? In particular, we're going to be sending out a survey. We really hope that EMGS members are ready to give back and be involved in this. I would say it's been really interesting working with the GTTC and these big collaborative groups to build the AOPs together. And there we're getting fantastic input from the DNA repair community and different making sure that the DNA pathways are described in sufficient detail that they're correct and they make sense, but not in so much detail that we've got an overwhelming amount of information. That we have the DNA repair key events in the right place and the right number of times, because it comes up so many times, and we're trying to figure out how best to do that. And so there's a lot of opportunity for the basic and applied communities to come together in this area. We've already talked about bringing the ecotox and the human health people together in this field too. So it's really, you need to think about people who can also who also study and understand the front end, the molecular aspects of the pathway, and then those who understand the back end and the sort of organism population level effects. So that also requires collaboration. And then we need the computational folks to come in and help us build the quantitative relationships. So there's many, many ways to get involved and many different areas that need to be reviewed. So we hope that you'll be interested. And
but I think it's important to be honest up front too, is that this isn't, it's a significant contribution of your time. Like the AOPs are, like we said, very large review papers. So I think why we want to make sure before we jump forward into this that we have the support from the membership and that they're aware of how much work this really is. It gets easier as time goes on, but it is an investment.
Well, that's why it's so important to do it now, because as we've developed these early key events and key event relationships, if they're developed well, then the review of those should be pretty easy as they're being reused. They should be updated. They should be, assessed for, do they still reflect the science in 2020? But technically they shouldn't need to be extensively rewritten or anything like that, right? So it should get easier as we move forward. And Bill asks, is there any money in this? No, the answer is there's no money to be had. You have to do it for the sheer joy and satisfaction of having given back to the larger community. There is some investment of time, of course, so it depends what you mean by money. I see a comment from Francesco. A comment needs to be endorsed by the OECD. It was not clear to me that endorsement that a specific process was followed, but does not cover content. Doesn't that limit the use of endorsed AOP by the regulatory community? So, I mean, yes, they've endorsed the process, but they have also looked at the content, and they're not going to endorse something that they really disagree on in terms of the content. But that's pretty much very unlikely to happen, given that an expert review community would have come together to have reviewed it. And you want to add to that?
No. I will say that one thing that I personally would like to clarify. I have that same question to Francesco, and maybe we can make that more clear what endorsement actually means. What are they saying? But
there's an actual statement that the OECD makes when they endorse them. We should pull that up and we can circulate that to the membership. Bill asks if this is valuable, who is paying for it? So who is paying for AOP development are the authors in and of themselves and their institutes. A lot of this work is being done by government institutes. And in terms of review, the initial review process was funded by the OECD, but it's become too big for them to fund. So if we made this part of the EMGS and the EMM, then essentially we're operating as under the journals framework, I think. So these are volunteer jobs. When we review for journals, this is a volunteer operation.
But you got to recognize that the goals of the AOP program are in alignment with the goals of the EMGS, and that's to bring the latest technology and knowledge and make it useful to regulatory testing. And this is one of the best ways to do it.
Yeah.
And so as a personal example, that's kind of my job. I think in my job description is to help bring the latest technology to chemical testing in Canada.
I'm not sure we've answered your question, Bill, if you want to unmute yourself and okay, well, if there are no other comments or questions, Jason O'Brien, EMGS member, you can find him in the directory, but he and I are both willing to talk to you offline about this anytime. You can email us with questions. And as I mentioned, we will talk further. We will hear feedback that you provide to us directly or through Bob. And we will talk this over again with the executive board and council and with EMM and decide how to proceed. And we will be sending around a survey to get your feedback following this. We'll give it a few weeks so that those people who weren't able to participate live have the opportunity to listen to the recording as well. And so with that, thank you very much for your time and for listening to us. And we're really looking forward to move. Jason and I are both very excited about this, and we're looking forward to discussing this with you over the next few weeks. Thanks. Bye.
Thank you very much. This concludes the EMGS webinar.